Dr. Sage here, continuing our discussion of viruses and prions. In today's video, we're going to discuss the general structure of viruses. By the end of this video, you should be able to discuss the size of viruses relative to other microorganisms, describe the function and structure of viral capsids, distinguish between enveloped and naked viruses, explain the importance of viral surface proteins or spikes, compare and contrast the composition of a viral genome to that of a cellular organism's genome, and diagram the possible nucleic acid configurations exhibited by viruses. So, viruses are smaller than the average bacteria. Therefore, electron microscopes are required to detect them. For example, parvoviruses are 0.02 micrometers in diameter. Mimiviruses are 450 nanometers in length, which is larger than some small bacteria. Cylindrical viruses are 0.8 micrometers long, but 0.015 micrometers in diameter. So here's an example of the relative sizes of things. Now notice this is a logarithmic scale. So what that means is every time we go up from one unit to the next unit, we're going up by a factor of 10. So we have things that you're typically used to thinking about, things like animal cells or plant cells, which are fairly large, or red blood cells, which is a type of animal cell that's a little bit smaller. We have organelles inside eukaryotic cells like the mitochondria. Now, similar to size to that is bacteria. Now, smaller than the majority of bacteria would be viruses, starting with the smallpox virus, and then smaller than that would be the flu virus, smaller than that would be the polio virus. So you can see, for example, that the flu virus is 10 times smaller than the average bacteria cell. So viruses bear no resemblance to cells. They lack the protein synthesizing machinery. They need only those parts required to invade and control a host cell. That would be the external coating and the core containing nucleic acids. The viral components include capsid, which is the shell that surrounds the nucleic acid. The nucleocapsid is the capsid and nucleic acid combined together. Some can have an envelope, so it's not found in all viruses. And in the envelope is usually a modified piece of the host cell membrane. They also have spikes, which are found in both naked, that would be the viruses without an envelope, and in enveloped viruses. They project from either the nucleocapsid or the envelope, and they allow viruses to dock with their host cells. Other virus terms is naked viruses. They consist only of a nucleocapsid, they don't have an envelope. And viron is a fully formed virus able to establish infection in a host. So if we look at generalized structures of viruses, they're going to have a nucleic acid. That could be DNA or it could be RNA. They're going to have a capsid protein coat around their nucleic acids. They're going to have spikes. While a naked virus does not have it, an envelope virus has a viral envelope surrounding the capsid. So capsomeres are identical protein subunits that spontaneously self-assemble to form the capsid. We have different shaped capsids. We can have a helicocapsid, which is rod-shaped capsomeres that form a continuous helix around the nucleic acid. A icosahedral capsid, which is three-dimensional, 20-sided figure with 12 evenly spaced corners. Or a complex capsid can have multiple types of proteins and take shapes that are not symmetrical. So here's an example of a helicocapsid, like the tobacco-mosaic virus, where the protein coat forms a helix, and within that helix is the nucleic acid. Or the icosahedral shape capsid includes the human rhinovirus. Looks like a 20-sided die if you're familiar with that. Or a complex capsid. Now let me give you another example of complex capsid. So a complex viral capsid, for example, can be found in bacteriophages. So these are viruses that infect bacteria. They have multiple types of proteins and shapes that are not necessarily symmetrical. As I briefly mentioned, we can have non-enveloped or naked viruses and enveloped viruses. So an example of a naked virus is the adenovirus, and an example of an enveloped virus is the human HIV virus. The viral envelope is composed of the membrane system of the host. It could be the cell membrane or the nuclear membrane. Regular membrane proteins are replaced with viral proteins, and spikes are protruding glycoproteins essential for attachment to the host cell. Nucleic acids are at the core of the virus. The genome is a full complement of DNA and RNA carried by a cell. Viruses contain either DNA or RNA, but not both. The number of viral genes is small compared to that of a cell. They possess only the genes necessary to invade host cells and redirect the host cell's activity. With the RNA viruses, we can have positive sense RNA, that's single-stranded RNA genomes ready for immediate translation into proteins. We can also have negative sense RNA. This is RNA genomes that need to be converted into the proper form to be made into proteins. Other substances you can sometimes find inside the virus particle could be enzymes for specific operations within the host cell. For example, polymerases to synthesize DNA and RNA, replicases to copy RNA, reverse transcriptase, which makes DNA from RNA. You can also have substances from the host cell within the virus particle. For example, rhinoviruses pack along host ribosomes, and retroviruses borrow the host tRNA molecules. Here's some examples of some common pathogenic DNA viruses. You can have DNA viruses that contain double-stranded DNA and an envelope. For example, the one that can cause herpes. You can have double-stranded DNA that are naked viruses. For example, the ones that can cause genital warts or the stomach flu. Or you can have single-stranded DNA that is a naked virus, and these cause respiratory tract infections. We also have common pathogenic RNA viruses, where you can have a double-stranded RNA and a naked virus, for example, rotavirus, or positive sense single-stranded RNA and a naked virus, for example, the one that causes the common cold, the rhinovirus, positive sense single-stranded RNA and an envelope virus, for example, the one that causes rubella, and negative sense single-stranded RNA and an envelope virus, for example, the one that causes the common flu. This was your introduction to the general structure of viruses. Until next time, this has been Dr. Sage.